ฮัลโหลโอเคเชิญนั่งได้เลยนะคะตรงไหนก็ได้ค่ะแล้วก็ด้านหน้ามีกลุ่มหนึ่งใหญ่ๆจะไม่เชิญเลยนะคะแล้วก็ด้านหน้าเชิญที่นั่งได้เลยนะคะก็ยินดีต้อนรับนะคะสู่ My Move Space แล้วก็วันนี้ก็จะเป็นจริงๆแล้วเป็นกระบวนการทดลอง Creative and Performance Project นะคะก็ Dancing Brain แล้วก็วันนี้จะประกอบไปด้วย2ส่วน2ช่วงด้วยกันนะคะช่วงแรกก็จะเป็นกระบวนการของการ prepare การเตรียมตัวในการติดตั้งเครื่อง EEG ให้กับนักแสดงของเรานะคะแล้วก็ครึ่ง2ก็จะเป็นกระบวนการทดลองการเต้นทั้งหมดนะคะก็เดี๋ยวขออนุญาตเริ่มช่วงแรกเลยกันเพื่อไม่ให้เสียเวลาก็ช่วงแรกเนี้ยถ้าอยากเดินเข้ามาดูหรืออะไรก็ตามสบายได้เลยนะคะขออย่างเดียวค่ะตอนนี้เนี่ยเนื่องจากว่าเรามีไลฟ์ Facebook ก็ขออนุญาตทุกท่านในขณะนี้ด้วยนะคะว่า so we have a live Facebook and a photo so in case nowadays I have to ask permission for everyone before I'm doing anything now so thank you very much for all your kindness and after that after the shows will be a Q&A sections also so please join us okay นะคะ
ได้ในกระบวนการนี้ครับแล้วก็เชิญทุกท่านเข้ามาชมใกล้ๆก็ได้นะครับถ้าสนใจ in this process we welcome everyone to kind of observe closely yes and then we would like to invite อาจารย์ชอนครับ to kind of perhaps share a little bit about the what is EEG and then how the brain create electricities ครับอะไรนะ yes
It's not okay oh cool ครับสวัสดีครับผมอาจารย์ชอนศิรวัตรนะครับก็วันนี้ก็ขอขอบคุณที่เชิญมาถอดหน้ากากอยู่นะครับ so thank you for inviting me today and briefly EEG is just a electrophysical sorry, electrophysiological signals that we measure on the skull you know like you know that you guys know what is the smallest part of our brain have you heard of neurons so neurons is เซลล์ประสาทนะฮะซึ่งเขาจะส่งสัญญาณประสาทเนาะไประหว่างเซลล์นะครับซึ่ง neurons เนี่ยจะมีลักษณะที่ยูนิคก็คือเขาจะเหมือนต้นไม้คล้ายๆต้นไม้ก็คือจะมีตัวเซลล์ถ้าเกิดมองมือได้มีตัวเซลล์แล้วก็ตรงแผงที่เป็นนิ้วมือเนี่ยจะเรียกว่า dendrites และตรงโคนเขาเนี่ยเรียกว่า axon ซึ่งนิวรอนหนึ่งตัวเนี่ยจะรับสัญญาณมาจากตัวที่เป็นกิ่งก้านที่เรียกว่า dendrites แล้วก็ส่งสัญญาณที่ transduce เขาเรียก signal transduction เนาะไม่ว่าจะเป็นแสงสีเสียงนะครับกลายเป็นสัญญาณที่เป็นในลักษณะเคมีไฟฟ้านะครับแล้วก็ส่งสัญญาณไฟฟ้าเนี่ยลงจากตัวเซลล์บอดี้ไปสู่แอ็กซอนเพื่อที่จะคอมมิวนิเคตหรือสื่อสารไปกับนิวรอนอีกตัวหนึ่งนะครับและระหว่างนิวรอน2ตัวเนี่ยbetween the two neurons there will be the small space just little space called synapse. Yeah. Synapse is where two neurons kind of talking to each other, right? And to talk to each other, there's these chemicals called neurotransmitter หรือสารสื่อประสาทนะครับคราวนี้จะเห็นได้ว่าตัวเซลล์ประสาทเองเนี่ยเขาสื่อสารด้วยกระแสไฟฟ้านะฮะคราวนี้ so if you look into the brain there's no brain picture here but if you look into inside of the brain there'll be the population of neurons known as pyramidal neurons that lay perpendicularly to the surface of the brain, right? So if you look at the cortex of เปลือกสมองก็จะมีตัวนิวรอนประชากรประสาทพวกนี้เรียงกันอยู่เป็นแนวตั้งฉากนะฮะเป็นแนวตั้งฉากกับกะโหลกของเรานะครับคราวนี้สิ่งที่เกิดขึ้นก็คือว่าเนื่องจากมีกระแสไฟฟ้าที่ส่งต่อกันไปเนี่ยนะครับก็จะทำให้เกิดกระแสไฟฟ้าที่หมุนเป็นวงจรเข้าหมุนนะฮะเข้าไปในตัวเซลล์แล้วก็ออกมาจากตัวเซลล์เป็นรูปลักษณะอย่างนี้วนอย่างนี้คราวนี้พอมีสัญญาณไฟฟ้าแบบนั้นแล้วเนี่ยมันก็จะมี electrical dipole ก็คือตัวกระแสไฟฟ้าเนี่ยพุ่งออกมานะครับพุ่งออกมาจากนอกสมองคราวนี้ถ้าเกิดเราติด electrode แล้วเนี่ยเขาก็จะสามารถ pick up small electrical signals ได้นะครับสัญญาณนั้นเรียกว่า EEG นะฮะ EEG ย่อมาจาก electroencephalogram electro กระแสไฟฟ้า enceph แปลว่าหัวนะฮะ graphy ก็คือกราฟนะก็เป็นกราฟกระแสไฟฟ้าที่แสดงถึงสัญญาณสมองนะฮะคราวนี้อันนี้เข้าใจไหมเข้าใจไหมฟังแล้วเข้าใจมากขึ้นอันนี้สดนะฮะสดเรียนมานานประมาณเกือบ20 ปีได้ If you want me to help I can help you too yeah so you can make for neuroscientists or psychologists right we interested in how the mind work and the Western feel about like the view of like the Westerner in terms of how we look into the mind. We start looking into the biological basis of it, which is the brain, right? So people might be interested in understanding how the brain kind of process all these signals that come into the brain. And how those are integrated and being encoded in the brain so that we can see whatever we're seeing right now, right? So indeed, whatever we see right now, me talking here, or like you trying to understand my conversation that I have with you, that's all processed in the brain, right? So there's this relationship between perception and the brain. And today, I think the team will demonstrate you how we could visualize what's going on in the brain while people are doing some type of action, right? And today, I guess the case study here is dancing, right? And if term dancing, there's this part of the brain called motor cortex, which is at the center of the brain. As a neuroscientist, we always taught, our students that there's this theory where we think that there's little human actually in the brain, inside the brain. It's called homunculus. Have you heard of homunculus? So it's kind of like this little human actually in the brain. Trying to control like how we are moving and stuff like that. And that is the motor cortex, right? That's the motor cortex. There will be a motor cortex. And what does motor cortex do? It's basically sending signals to the effectors, in our muscles, So if we're thinking that we're going to move the left hand, then the right motor cortex would spark, you know. And the signal will send through and cross over in our spinal cord, and then send it over to our effectors so that we can move like this, right? So right now, my motor cortex is spiking, right? So if actually, in addition to EEG, there is this technique called TMS. It's called transcranial. Yes? Yes. Yes. Oh, yeah. So to be able to pick up very small signals, right, you have to have the electrode that have a very low impedance. If you learn in physics class, right, V equal IR. So is that right? จำไม่ได้กระแสไฟฟ้ากับ resistance ใช่ไหมฮะ V equal IR V คือ voltage เนาะI คือกระแสไฟฟ้า R คือ resistance and that's resistance is the impedance. So right now what they're doing is basically filling in the gel which is an electrolyte. So this gel will decrease the impedance of the electrode. So you can see here, right? This number is what? 200 kilo ohms. That's the impedance. So right now, if this electrode, you can see that it's white, and it shows INF. INF is infinity. So the impedance is very, very high, right? It's very high. So in that case, you won't be able to pick up the electricity, the current, the I, right? Because given that the voltage remains the same, right? If your R is resistant or impedance is high, then you're not going to have the current that flow into the electrode itself. So what you want to do is put in the gel and make a contact, right, between the skull, which is the skin, and the electrodes. Right? So as she's filling in the gel, then you can see that the electrode itself will turn from white to yellow and then green. So green would mean that it's very low. So it's very low on the other end. So I guess it would be like 10 kilo ohms, something like that, which is a standard that we use in experiment. Actually, if you're in a very strict lab, we would like it to be like below 3 kilo ohms. So in that case, we will have a lot of signal that's coming into each of the electrode. And this is a caveat. While filling in the gels, you actually need to make sure that it's not connecting to one another because you want to separate all the sources that pick up all those signals, right? So as you fill in, you want to make sure that the gel is kind of like restricted in a certain area so that it can pick up, about 2 or 3 centimeters of space that contain, those neuronal populations. Is that clear to you? Thank you. So, I lost my train of thought. What else should I talk about?
I think right now we try to discuss about. For recording the data from the dancers, we're not only collecting the brain wave, but also we are also collecting the muscles, activities, and the accelerometer as well. And then why is it important?
If you're interested in behavior in the brain, definitely you have to get some type of behavioral measurements you know. And this team is so smart in terms of coming up with, you know, how you integrate those measurements, right? Before you integrating them, you need to measure them, of course, right? And by putting the electrodes, this is called EMG, I think. I guess you're gonna put like in the back of the dancer, right? Back and lower back, so that you can kind of measure the movements
Yes.
using the electrodes as well, you know. So you can do that on any given type of muscles. Usually in the experiment room, not dancing, like when you having like human subjects in front of the computer and they're responding something like playing games, something like that. What you can actually do is putting the EMG electrodes on the fingers, on the fingertips, another finger, on these muscles, this muscle, you know, on these two fingers. And then when subject try to respond, you can actually measure muscle activities. And I was gonna, before you interrupt me, I'm sorry, I'm kidding. Before you ask the question, I was gonna introduce you to this method called TMS, which is transcranial magnetic stimulation. We were talking about the motor cortex, right? Being the part of the brain that sends signals so that you can move around. Instead of thinking about moving, what you can actually do in experiment room is putting stimulation, like magnetic stimulation through the skull, right? So if you want a certain people to move, let's say their left hand, you might want to stimulate around here on the right side of the brain, central part. And when you stimulate it with a magnetic stimulation, then it would induce this electrical activity in motor cortex by just stimulating it, right? And by doing that, actually people will start twitching like this, because stimulating it, sending the electrical signal to the muscle itself, right? So these techniques being used a lot to study how we human stop our action, right? Let's say if you're on diet and you really want to eat hamburger or something like that, right? There's like big research group that look into like how we actually, how the brains actually send signal to kind of stop our urge or something like that. There's a whole big field of study looking at this kind of response inhibition. And that is really important for clinical implications, like how do you help people with addiction problem, how you help people with eating disorder problems or something like that. But today, the team's not gonna do that, but today we're gonna, step outside the box a little bit, I guess, and see how we can kind of combine, brain science and arts, and how we can visualize that so that you can see, what may be going on in the brain while people are dancing or improvising the movement or even thinking or imagining about the movement. Yeah. Yes. Yes. Yeah. Yeah, how do we manage that? Yeah, there's a technique called ICA or PCA, which is like a machine learning method to kind of learn the source of the artifacts, right? So that is very good question, by the way, because we have to deal with artifacts a lot when we measure signals like EEG, which is very small, You know, so even the small eye movements can interfere with what's going on in the brain, in terms of how we picked it up. So we have this method called independent component analysis that we use in the lab. We use in the lab to kind of see what type of signal from what sources, you know. So let's say like you have all these electrodes, is 64 electrodes, right? So basically here you have 64 independent sources, and it would pick up all kind of signals that project onto that electrode, right? But by using such analysis to kind of see how are they picking up different signals independently from one another, you can kind of sort that out and then see, okay, this signal looks like eye movement. This signal looks like head movement. This signal looks like people crunching their teeth or like raising their eyebrows or something like that. And when you look at the variance that explained by each of the component, you can pull it out and just reject it. And by doing that, you will get like very clean signals. Although there's still people in the field using different kind of method as well. You know, like some people might use some techniques, the very traditional techniques called threshold rejection. What you can do is when you have head movement or eye movement, something like that, you can just like have the hard threshold. And if the EEG signals go above or below the threshold, you say, we're gonna reject those trials because we're interested in the brain. But in the experiment like this, you actually cannot do that because subjects are moving all the time, right? So you lose all the signal that you want if you use threshold rejection. That's why techniques like ICAs or PC would be important and necessary in this type of experiment. Yeah. In real time. Yeah, you can do that. I'm not sure that are you guys implementing ICAs here? Probably not. Yeah, it requires a certain time to do so, right? Because you have to compute, you know, at every time point, you know, what's the independency between all those signals, right, from all different electrodes. So the more electrodes you have, you know, you might have to spend more time computing it, or the higher sampling frequency that you have, you spend more time on it. But the team actually at UCSD, University of California, San Diego, there's this team led by John McKeek and the other guy's name, Chesnowski, the guy who write deep learning book. They came up with a faster algorithm that you can implement at real time. Yeah, you just have to kind of figure out how to implement it in this kind of real-time setting. But it's definitely a challenge, and you have to have the experience of doing that. I myself never done that. I've done fast ICA, but I haven't done in real time. No. Yeah. Thank you for the question.
Right now we are kind of connecting the muscles activities, and then maybe why we choose these three spots. Maybe Pimam can elaborate a little more.
So what we try to do now, in fact, this project starts from my craziness idea of my study about the aesthetics of dance, especially ballet. Because ballet training, all the ballerina have to be perfectionists. You have to know how to control your muscle. You have to know how to control your body just like exactly 100% where it should be. Just like my last research was about the connection between ballet and mathematics. Because everything has to be precise. They call each part of the body as a dimension, as an angle, as a direction, just like on the floor, 45 degrees, 90 degrees, up above, from the semicircle to the circle and everything. It's just like you have to learn how to be precise to make it beauty. So body makes something like muscle memory. we call in the training, we call muscle memory, because you keep training that from 4-year-olds coming up, maybe 20 years. And our question is, when you want to dance perfectly, what happened to your brain? How is your brain dancing? Is that a memory? Is that a control? Is that a coordination? Especially why we all need you here is, especially when you have an audience. Is that different? So I believe that every time the brain muscles working different, the brain work different, the muscle work different, but you have a basic training. So all the muscle that we put it on is the main muscle that we use for dancing, which is the inner thigh, the lower back, and the upper back. So that's it all main. We can't, but with the limitation of course, this is from our lovely sponsors. So with the wiring system and all the heaviness of the machine is limited a bit of the training. So what you will see is a set basic exercise that every dancer doing it since 4 years old, 5 years old coming up, and it's all set. And we've been just like experimental and collect the data from last whole 6 months, but then now we try to do it with the audience and see how it's going, how it's different.
Yeah, and what Ajahn Mam just said is actually what people in the field of neuroscience, focus on, the learning process. You might have heard of neuroplasticity. Our brain, what is plasticity? Plasticity is like plastic, very flexible, changeable. Our brain, learn at every given minute, and it's changed at every given minute. Even like with people who's older, right? We know that inside of the brain, there's a place called hippocampus, which is important for memory. There's a cells being generated across our lifespan, right? With dancing, it's really interesting, as Ajahn Mam raised earlier, right? That could be, the dancer could be rehearsing, right, without the audience, the cognitive state would be totally different, when you're just rehearsing by yourself versus, have this audience. The load, the effort that you might have to use, or even like what type of memory or learning that you're using could be different. Procedural memory or muscle memory, or something that episodic, semantic, And this involves different type of brain processes. And it actually would be really interesting. I think this research team are looking at, different types of learning and behavior, like the dancer rehearsing, or improvising, or even like imagining herself dancing could evoke, induce, could evoke different kind of neural processes. Yeah, I guess you guys are ready pretty soon. Yeah.
Okay.
Yeah, thank you.
So after this, what you will see is another 30 minutes of process of trying to dance. And in a set routine, swap with just like a less eye open, so you can see the different images of her brain while stay still, open your eye, close your eyes, and dancing, and of course improvisation. Every visualization that you saw, you will see is from a live data processing from the brain. So it's different, and then we come back and talk later then after finish, okay? Enjoy.
Okay, ready? Rest eye open. Rest eye close. Dance 1. Rest eye open. Dance 2. Press eye open. Motor imageries. Rest eye open. Dance improvisations. rest eye open.
Okay, so we are at the last part of our
ขอเชิญพี่ชอนด้วยครับ
Mimi โอเคค่ะ So let me before I start everything อาจารย์ชอนเชิญค่ะ let me introduce our dancers a bit. Her name is Mimi Wararom. And this is her place, Mind Move Space. This is her own studio. So I have to thank her for to let us doing everything so here with super experimental equipment like this. And okay, my three brothers still stuck with our programs a bit over there. Okay, so this project, in fact, start with me and Henry who get together, and Henry used to do some project with the brain, kind of a meditation system, while I did a research with the aesthetics of ballet and measurements and all the kind of linking art and science together. That is my whole idea of my my personal research. And this project, in fact, is supported by Chulalongkorn University, Faculty of Fine and Applied Art. We have a center called Fine and Applied Art Multidisciplinary Art Innovation Program which support by C2F กองทุนศตวรรษที่2นะคะ Sentinel Fund it is a center that support digital system, digital art as a tool for the artists in variety kind of presentation. So you can use a digital art in any way as a tool to present your idea, either visual arts, fine arts, mixed medias, new medias, movie, dance, music. And our next project is the literature, the wording with the AR. That's coming again in September. But we have the new, let me promote this a bit, okay? Sorry. We have a VR exhibition starting next week at our CU Art 4C in Palamsi. We have a small gallery over there, and that artist that we collaboration with the Digital Art Foundation Taiwan. So they come from Taipei, which is a 3 exhibition, and that will run for 4 weeks over there, but you have to book because it's very interesting. We are 360 movie, a dance movie, and that's 20 minutes long, but you have another 3 pieces. So please stop by, and I have a bit of information and a Facebook for you to follow. So mainly for us, we believe that art can't speak only one language anymore. Nowadays, it last few years that art been linked together with science. You will see a new trend that coming this year. I think only more and more this year. It's called bioart. C-Lab is just like a scientist and technology labs and everything coming up, which we're going to have a talk about the digital arts in Taipei and Thailand on this Tuesday at 2 p.m. at BICC, which you are welcome. So we will talk about all these changes in art social and science socials, how these 2 things can start to matching, and we do a lot together right now. I think this is the whole idea about behind the scene of this project that's going on right now. Henry, you want to add on your side? Anything else?
I mean, how about we discuss a little bit about
Yeah.
what's going on, and then what do we see, and then or if you guys have any questions, even technical questions, then we're happy to share or discuss about. How about Chan Chon? What do you think?
After seeing all this, what do you think?
It's awesome. I think I just post on the IG. It's like a really freaking cool idea, not to be crude. You know, it's just amazing to see. You know, like I'm a neuroscientist, so I'm in research lab all the time, right? And you know the hardest thing I would do in my career is just going to the conference and giving presentation, right? And definitely what we struggle in terms of like being a scientist is convey those messages to the people, outside our field, how to communicate the science better. And, and today I came here and see, the magic of art and visualization. And the most important thing is like, how do you bridge, the science, brain science into art? That is such a cool idea. And yeah, for that, I applaud. I'm just speechless. So I applaud this team a lot for making this happen. And I'm glad to be part of it. Just a small part of it. Yeah. Yeah. Yeah.
Thank you for that. And then also how we come into interest in different activities of the brain while Mimi is practice dance, and we also thinking about what's going to happen inside her brain while she was imagine a dance, and then we found sometimes it's quite difficult for her during rest eye open more than
the dance.
More than imagining the dance.
For someone who doesn't know, you might notice the color of visualization I didn't tell you before. Mainly it's red, yellow, and blue. So we use a spectrum to transform the data so you understand more, easiest. So if the brain work just like heavily, just like thinking a lot, it's going to be red. Something like that. If the electricity going on just like craziness, it will be red, totally very red. So every time she dance, as a dancer, I tell you yesterday worse, today is fine, okay? Because we keep looking at her brain for about last 6 weeks. So I noticed as a dance teacher, a dancer myself, I noticed something because if you notice, the exercise and the bar at the beginning, the first and second dance is the same. It's totally the same. But as we start on different side, the first one we start with she facing your audience. The second one starting facing backward. It means she used different side of the leg in each one. So the hard part will be on different side. And of course, as a dancer, athlete, spotters, anyone, you will have a good side and bad side. And unluckily, because I want to use a mirror as a part of the design, so she have to facing the audience on the left side, which is her bad one. So every time, everything, the hard stuff, which is all the lifting stuff, all the kicking stuff on the side, her brain will be totally very related on every hard exercise that she have to control more. And every time that is just like have to train control, you will see just like today, just like this is a technical term. When she do a combo on the second side, again, that one, when she kick, it's totally red or yellow. So you will think that, you will know that they will just like something that is working hard, which I can tell you now. Yesterday, her teachers who teach her since she was 5 years old sitting over there on a very bad angle for the dancers. So it's totally yellow and red nearly all the times. But today, I think she feel fine because I saw your brain was like totally very blue on nearly the same times. Okay.
But I mean, more on that is that when the dancer is study the dance, the mirror neuron is more activated when how you learn the dance, because we can't learn the dance by reading. We learn the dance by seeing how our teachers teach you how to dance. And then when you are performing the dance yourself, the motor neurons would be more activated. And then for the dancers who've been practicing for years, I'm quite questions how the brain is working or pull the memory out. Is it from which part of the memories that the brain is using, or is it instinctively? What does it mean instinctively in terms of how the dancer is kind of remembering and rethink of the dance movement? And then this is a first part that kind of we are so curious about this project. And then for Mimi, perhaps you can share a little bit about how you, I don't know, is it your body move it by itself when you remember this dance, or you have to think about it?
I guess that's a research question.
You want to open that one again?
It will be totally red now. So, okay, I have to confess, sometimes I just dance instinctively, and especially at the bar, because the exercises were set, and I kind of remember by my body, and sometimes not today, but some other days that I felt very tired and I felt as sleepy, and sometimes I forgot where was I, and yeah, I didn't think much in my brain about the steps, but when I'm doing something on my bad side, the difficult stuff, I will think a lot in terms of technique. I should pull up my supporting leg more. I should point my feet more, something like that. That's small details. So I don't know I can answer that question or not. It's difficult. That's why I have to wear this one.
I also think that when you have audience, it's totally different. Because if you're just like, Mimi was a professional dancer in Singapore for 7 years. So when you have dancers, you have audience. A dancer, you have audience. You got a kind of pressures. You got a kind of bit feelings. So I think that from our data collecting, testing, until just like with the audience today is totally different.
And when you talk about the audience, right? It's when we collected the data before today, I felt more, of course, more relaxed. But today, when we have audience, like yesterday, I also said this one, I felt self-conscious and I felt a little bit judged. And yeah, I think it's normal. So what you see in my head, in my brain, on the screen, maybe it's not that I'm thinking about how I would perfect my step, but it's about my being self-conscious or being a lot feeling, I'm not slim enough today or cellulite on my legs. So that's the thing.
And John Sean, perhaps you can elaborate a little bit more about, like, for example, when yourself is maybe doing a workout or some sport, do you?
Yeah, definitely. You know, like the brain, like is modulated by the arousal states, right? So like you can go into the situation when you're more stressful, something like that. It would induce, you know, different state of the brain. You have more arousal, then you have very high alpha activity or something like that, and that would, you know, not only impact you know, how the brains work globally, but also at different parts of the brains are being impacted by arousal states. So we have studies that look into how stress impact activity, like let's say visual cortex. Studies have used actually mice, like a little rat, to run around, and actually see that when you when the rat is like fearful of something, the locomotion activity in the motor cortex are so different than when they are not. So that's how stress would impact the brain, right? So what you might see is probably a combination between, you know, the arousal states that impact the brain and also, you know, some of motor activity. You know, there's a sound, so there could be like auditory activity as well. And as you move, your head is moving, and you know that could also be visual activity. And like you suggest earlier, it could also be artifacts as well. So you know, you have to be concerned that, you know, in experiment like this, you know, it's important to kind of tease apart what's noise and what is real signal, stuff like that. Yeah, and I really appreciate what you were just mentioning earlier about, you know, different manipulation that you do here, right? You have the subject, human subject, you have a professional dancer, you know, rehearse, you know, from muscle memory versus, you know, imagining it, right? Versus when she's using very hard, you know, not well practiced, what do you call that? The side that she's not familiar with, then no, she would like bring out other stuff from her memories, and it's different from of memories that can actually interfere with one another. So let's say when you're younger and you're trying to learn to bike, right? If you use the part of the brain that's like, okay, how should I bike step by step, step by step? You wouldn't be able to bike, right? You would have to fall down, you know, and learn from try and error to be able to bike. So this kind of experiment is kind of showing that there's two different types of memories, and they are interacting, inhibiting one another, something like that. So if you can go back and look at the EEG data that you're collecting and try to look at neural signals that correlate with a certain type of memory, that could be really cool. And I really like, this is just my comment on top of the, you know, so the art show today, I really like that it's very experimental based as well. you know, the way they did is like, you know, you have to go back to the baseline with eye open, you know, eye closed, so that, you know, we know already like what's going on there, right? And then compare those baseline to what activity you have the dancers doing, you know, improvisation, the dancing, the rehearsing, and also motor imagery. And what we could later on do is trying to, you know, analyze it in many different ways, you know, just not just look at the brain wave, but also maybe you can do some type of similarity analysis, you know, whether you can use machine learning to see, you know, when she's imagining things versus when she improv, when she's rehearsing, are there signals that similar for one another or not? There could be so many implications that come out of this, yeah.
And then also the last experiment that we tried to we interested in is that how brain is working is why we improvise, and because I'm also sport players, and then sometimes when we have to make decisions at a split second, and then sometimes I'm not sure if I'm actually thinking or it just how it works. So, and then also if it's also the same as dancer as well. So how when she's not preparing what she have to dance, and then when she have to think on site as improvisations, what is going on. This is I'm very curious. Maybe Mimi can share a little bit of how you think about your choreography while improvisations.
I also want to see my brain when I was improvising because, yeah, I don't know about today, but the days before in the earlier rounds, it's mostly blue, right? Yes. But I don't know.
Yesterday it was disconnected.
Oh.
The cable was disconnected because you danced too big.
May I tell you first? Our big, just like a big problem here is the machine didn't design for dancing. You have a lot of wires, and it just like and the mesh. And when you do ballet, it's from head to toe. It's every corner is moving. You keep moving all the times. When I do it myself, because we start trying with me, I'm dancing myself. It's lost from just like this part, just like from the very joint connection, because it's limit yourself a bit. So the choreographing that I did is the best I can do. I can choreograph for her that the machine, and she still can do it. But with the wires, sometimes it's not. So it's sometimes that we dance for 1 hour, collect it, and then nothing. It's happened so many times with us.
Yeah, because sometimes like contemporary dance, you have to roll on the floors, or you lie on your back, or you have to kind of roll your necks, and then there's always a limitation of the device. But yeah, but how your brains work while you're
While I'm doing improvisation, I felt quite relaxed. On a normal basis, I felt quite relaxed, and I let it go. Whatever comes, I'll do it. I'll let myself do it. But when Henry talked about the split second of making decision, and then I thought, yes, I did a lot of that. Like, should I step on the chair, or on the stool, or should I sit on the floor, or should I run to the bar, or should I run around? And yeah, there are a lot of those decision making going on, and I think that's why it's red, maybe, because of that. And like Ajahn Sean said, it's about my memory. I don't know. The nickname is muscle memory, and my body remember how to do it. So I kind of, sorry in Thai, like ตกลงไปในร่องเดิมอะค่ะ Yeah, like I follow the same steps all the time. Like, oh, I did it again. It's like, yeah, it's my kind of my signature, that kind, and I did it a lot, and when that happened, I think that's why it becomes blue. When I try to step out of the frame, okay, I'll try something else, and then I have to make a decision, and that's why my brain works.
Also, at the end, like the EEG machine is like itching or something? Like you're just like scratching because it's itchy? I'm kidding. I'm kidding. That's the limitation of the machine, right?
Yes.
But talking about like the machine itself, I'm thinking about BCI. Have you heard of BCI, brain computer interface, right? So indeed, like what they're doing today, model imagery is actually one of the techniques that we use in BCI for what? For helping patients, you know, who cannot walk, who cannot speak, or something like that. And you combine that with machine learning to kind of decode what, you know, the patients are thinking so that you can, you know, build applications or help them with rehabilitation, you know. So, you know, for today, what you see is not just for rehabilitation anymore, right? You can use arts, you can create arts. Let's say if you want to connect with the VR system where the dancer in the virtual reality is actually dancing along what she's thinking, right? And you know, by doing this type of experiment, that could happen, I think.
Now this part, I would like to share a little bit about the design of visualization design. So, I mean, we basically use the scientific program pipeline. We use the Ego Sport software, which inside this laptop at the back. Then we kind of send the signal wirelessly using the LSL streaming system to these computers. Then we use the MATLAB to collect the data, real-time data. And then one machine, we use the MATLAB to visualize the topography of our brains. And then another one, we also use the MATLAB and then feed it into the EEG lab program, which showing the real-time graph over there. Then we also use the program called NeuroPipe, which help to visualize the real-time informations to show the connectivities and the estimations. And then of course we also try to imagine how can we use or how can we propose the artistic visualization by showing the trees. So we're thinking about what if maybe an alpha wave is the blue trees or a beta wave is a red trees. Perhaps she can grow trees with her own brainwave. That's one of the proposals that we try to explore and experiment. And then maybe Ming can elaborate more.
So some of the visualizations right now, to be honest, they don't really tell you what Mimi's brain is currently doing. It's just extrapolation of brain activity. It's just telling you what parts of the brain are active or what parts of the brain are being utilized. So the visualization is trying to do that. There are quite a few problems with, as you said before, noise or interruptions from other stimuli into the brain. So that is quite a significant factor in the computation of the signals, and trying to filter those stimuli out do cause quite a bit of delay between the signals. So yesterday it wasn't this bad, but today you would see like a little bit of desynchronization between the projections on the floor and the projections on the wall, because some of the algorithms that we use for the floor, even though we are using small algorithms and we're trying to make it faster, even though less accurate, are kind of lagging today. And like it's a really interesting problem because like you're trying to filter or analyze data that is being generated live. So it's like interesting part of the pipeline.
Yeah, now it's time for opening questions from the floors or any feedback or response, comments. Welcome.
Yes.
Hello, I have two questions. Earlier, I was going to ask, because Professor raised an example that you can on the contrary, reverse way, you can manage, you can stimulate, use the electrode to stimulate the human brain to generate human body's reaction, right? Like, I'm curious about how far it can go, how much currently the frontier science can do to manipulate humans' reaction. Can we stimulate certain part of the brain to let a human dance, to raise a leg? I mean, to what kind of extent?
Do you want me to answer?
Yeah, maybe.
In terms of stimulate human to dance, I don't think we go that far yet because, like, you know, let's imagine, like, you know, the most that you can do to, let's say, motor cortex is doing TMS, and what it's doing is like cause twitching, either like on the face or on the arms, something like that. And you have to imagine that you need to stimulate multiple parts of the brain at the same time so that you have this kind of complex movement. That's interesting idea. And actually, I don't think that anyone have done it, even in the mice. But in terms of technology to kind of manipulate like movement of an organism, there's indeed this new technique called optogenetics. So what they did is they infect the virus with, you know, they do it like genetic engineer so that part of the brain would express receptor that's sensitive to light. Right? So you might infect this virus like into the motor cortex, let's say. And then what you can do is you can put the LED light and do the surgery into the mice motor cortex. And what you can do is when it's running around, you start to, you know, turn on the light in the brain, and the brain will respond to the light, right? And you can stimulate that, you know, left, right. And if you go on YouTube and type optogenetics, the mice is actually turning around the way that we want, right? So the science of frontiers has gone that far. And this is like 10 years ago, right? So in terms of what we can do now, right? With brain stimulation, you know, we have in Parkinson's disease, we have deep brain stimulation. We're actually going to the brain using like this electrode, right? And today we learn about oscillation, like synchronization. What the surgeon like to do is just put the current that oscillate at a certain frequency called beta band. And that kind of helps sync the brain, different parts of the brain, and Parkinson's patients who have problem with tremor or problem with walking, they won't have those problems anymore with brain stimulation. That's kind of like how far, you know, the simulation technology is kind of go. Yeah, and we in the lab also use brain stimulation to help people learning better, to pay attention better, and have a better memory as well. So that is still on progress. Yeah. But not dancing. Haven't seen. It would be really cool.
Anything else? One more? Okay.
Sorry, I was just curious about another thing. Like, have had anyone tried to put the VR on the model? Like the model see the reality inside of the VR, how the brain function and reacts compared to when the model sees the real reality. Does it make any difference?
You mean like the VR?
You mean metaverse kind of?
Yeah, there's a lot of studies looking into that. I mean, in terms of VR, what like people study like depth, you know, the depth, like how the depth vision work. You know, that's been studied for many years. And actually, VR technology, we have VR technology for like 20 plus years. But it just get more popular these days just because of technology and because of Facebook. I want to change to metaverse, right? So indeed, like, you know, in real world, we use more part of the brain to navigate through 3D versus just sitting in front of computer. You know, in real world, not only we see it more depth, but when you go through the world, you're actually moving your head, right? You're moving heads, you're moving your eyes, right? So, yeah, it's using like a lot. I want to put it simple. It's using a lot of more part of the brains, and the important part is like how you integrate those information so that you know, you can navigate through 3D environments. Yeah, but it's actually, it's different, but I don't think it's that different. I don't think so. Yeah. It's just the way that we study. Yeah, human cognition used to be like on, you know, just a computer screen, flat screen, and just now that we start to do that. Yeah, there's a limitation in terms of using EEG or MRI to kind of study 2D versus 3D also, because we don't have much resolution, like in mice, where you can go through like, you know, different part of the brains and look at neuronal activity. So there's still a big gap. So there's no conclusion that I can make here, but yeah, that's what I know.
Thank you.
Another one question from.
Yeah, actually, that's kind of interesting because with VR, you know, you had that problem of latency that the body feels like it's slightly behind what it's seeing, and I think in some ways that relates to the gap with in a different way with muscle memory, which is really fascinating. What you were saying about how that relates to intuition and improvisation, where you're making decisions, and then when you're really in that flow, kind of the body makes the decisions for you, right? Whether it's coming from habit or if you're really feeling in the flow, it might just be a spontaneous thing that you never even thought. And so I was just wondering, I don't know, because you said that was maybe a blue state, but I'm just wondering to what extent you can actually measure those things, because I know from, you know, that idea of the half-second lapse between the body and the perception of what's happening now in the mind, and especially to do with arousal states. So if you're really like in a fight-flight kind of state, then your body is often reacting before you have a chance to really calculate what decisions you're making. So there's an interesting gap there. I think that also relates in a different way to the data gap and that problem of artifacts. I don't know if there's any way of, if it was ever possible to correlate those two things, of firstly separating out the different signals and then finding some accuracy of how the dancer navigates that space of intuition. That would be, and if you could model that would be really fascinating, but I don't know what the limits are, especially in real time.
I was thinking about that because when we first start, we really don't know what to do because, you know, just like as I mentioned before, the dancers move the whole body, really the whole body. Because the machine was starting making it just like this, especially this, just like movable stuff, they make it for the runner. So you keep repeating the step, and you just measure all the brain data by it's repeating the step. Another case that I found out is a bicycle, and everything do it in a lab. So you running on the machine, you bike on the machine. You didn't even go for the real weather, just like up and down the mountain at all. So with the dancing, I don't know, this is the first try, really, really the first try. So we still found a lot of limitation, just like, I don't know, even we have to go back to just like only doing a plie, the bending of the knee 100 times until we really know what is the function. So this is what our decision, that's why we want to see a big comparison between a technically setting up, so you got your brain muscle memory already, and then between the improvisation. So you might notice, and we might can analyze something that you see the big difference between. But just like if you want to think about that is a decision of doing something or not, I think it's even another factor, because if someone just like very used to be a professional dancer, and you saw the audience, you will play with the audience reaction. That also another input. And it's also with the props, with the stage, with the lighting, every single thing is changing. But that a nice thing, aesthetics wise for the improvisation. But there are also some type of the dancers who just let the brain go. Whatever, just let the body go. You hear the music, you got the atmosphere, and just have fun with it. There are also another type of dancer who did that. Just like if it's me, I'm going to be just like, okay, let it go. Do whatever you think and let the body go, because you've been training for that. And do whatever your dance, your body give a variety of movement. So I think many just like factors for that, to go that far. But I just like, will say that this one is only just like a pilot project. Really, really super pilot project to be able to start. Because I was thinking there was someone in our faculty of sports science who, when we start, they even told me just like, maybe shall we start only that first, and then go to the arms, and then go to I said, okay, if you think about that, the muscle go totally different angles from what I did. Because the first time I test, I'm only do the basic stuff from first to fifth position. But then data loss. So we don't know what is going on after that. But I think that's why we want to set up this today. So we know how it's going, and I hope from this data today, these two shows, we can start doing something with it. Because if not start, it's nothing we doesn't know how to going on after this at all. I hope this answer your question a bit.
Yeah, thank you.
Add a little bit. So like, what they're doing is not just like visualize, analyzing data at real time. You can go back and see and analyze the data. And what's good about EEG is that it has very high temporal resolution. It has millisecond resolution. So what you can do, given that they're measuring all the muscle movements, and all kind of behavior measure, video recording, sometimes people put like the white crayon, so that you can kind of image like how each limb was moving. And if you can go back in time and time lock those events, right? So then you can see how the brain activity are actually changing, right? And you can use past studies, the established neural index like motor, visual, auditory, something like that, to kind of see how they change and how it correlated with the behavior, whether the dancer are being precise or not, or how risky they want to take in terms of, improvisation and those kind of stuff. So there could be something that we can track back and then get the insight from it, yeah.
Okay, I think it's about the time for the live, because we live on the Facebook, and we have to clean up the space and giving her back to the space tomorrow. So I think Henley can go on a bit.
Okay, so I'll wrap up today. So thank you everyone for coming to the shows, and thank you Ajahn Chon Sirawat Iti Puripat for kind of advice for this project. Also, thank you Pongsakorn Vichakarn for also giving special advice for our research. Special support thanks to คุณกัญญรัตน์ คุณปัญชลี คุณประภาสิริ คุณณัฐพัฒน์ over here, and also thank you lighting designers คุณภริตา คุณครองชัย แล้วก็ technical designers คุณจิรัตน์ over there. Special thanks to ฟ้าใหม่ special thanks to Neuromaze Laboratories, Learning Institute, Kim Mongkut Institute of Technologies, Thonburi. Thank you to University of Thai Chamber of Commerce for supporting Ming here. Thank you to Faculty of Sports Science, Chulalongkorn Universities for letting us use the device for trials. Also, special thanks to BJC Healthcare and Ant Neuros, Piso, Pinan, and Pijop for supporting us for the past month and a half for technical errors and all the tools and equipments here. This is the device that giving from BJCs. And also thank you Mind Move Space Mimi for providing us big and spacious space here. And also thank you 4M Multimedia for providing us two very nice projectors. And also that projector is from Famai. And of course, thank you everyone for coming to join us today, and then hope you guys enjoy the show and looking forward for our future experiment.
Yeah, so we have Pham Mai is Facebook, Pham Mai Digital Arts Hubs. So it's mostly with anything is not only a project, anything that happened around in Bangkok with the digital arts or any interesting challenge in arts, you can follow us over there.
Last but not least, if you're interested to be a volunteer as a research subject, we have a poster post in front of the entrance. This will be part of the research from Neuromaze Laboratories. You also get paid. Yes.
Okay.
Okay, thank you everyone.
Thank you very much. ขอบคุณค่ะ